Hi friends, I am Anaga Srinivasan, first year MSc Biology student. Here we discuss about role of chemokine in management of AIDS. Let's see what is chemokines. Chemokines are superfamily of small peptides, most of which contain 90 to 130 amino acid residue. They selectively control adhesion, chemotaxis, and activation of many types of leukocyte population and subpopulations. Primarily, it is involved in inflammatory process. It plays important hemostatic and developmental roles to play regulatory roles in development of blood vessels and wound healing. The inflammatory chemokines are typically induced in response to infection. Chemokines possess four conserved cysteine residue CC subgroup, that is chemokines in which conserved cysteines are contagious, CHC subgroup, chemokines in which conserved cysteine are separated by some other amino acid. The core receptors CHCR4 and CCR5. The CHCR4 is expressed by neutrophils and CCR5 is expressed by Th1 cells. Let's see the steps in HIV infection. Viral attachment and entry into target cell is the first step in HIV infection. HIV-1 infects T cells that carry CD4 antigen monocytes and other sites have CD4 on their surface. Virus binds to CD4 on cell surface. A co-receptors present on T cells and monocytes required for HIV-1 infection. Infection of T cells is assisted by chemokine receptor CHCR4 and analogous receptor CCR5 function for monocyte or macrophage. Steps in HIV infection are represented as flowchart is shown in screen. Let's see. Virus entered a cell, then occurs reverse transcription of RNA genome of virus. The provirus integrates into host genome, that is integration of provirus, then transcription of integrated provirus, then spliced various viral RNA messages. The last step is RNA is converted into protein by translation. CHCR4 and CCR5 serve as co-receptors for HIV on T cells and macrophages. That is, T-tropic strain uses CHCR4 and M-tropic strain uses CCR5. Here the figure shows steps in HIV infection. Let's explain the steps one by one. HIV glycoprotein 120 binds to CD4 on target cell. Fusogenic domain in glycoprotein 41 and CHCR4, a protein linked receptor in the target cell membrane mediate fusion. Nucleocapsid contain viral genome and enzymes enter cells. Viral genome and enzymes are released following removal of core proteins. Viral reverse transcriptase catalyzes reverse transcription of single-stranded RNA, forming RNA-DNA hybrids. Original RNA template is partially degraded by ribonuclease H, followed by synthesis of second DNA strand to yield HIV double-stranded DNA. The viral double-stranded DNA is then translocated to the nucleus and integrated into the host chromosomal DNA by viral integrase enzyme. Transcription factors stimulate transcription of proviral DNA into genomic single-stranded RNA and after processing several mRNAs. Viral RNA is exported to cytoplasm. Host cell ribosomes catalyze synthesis of viral precursor protein. Viral protease cleaves precursors into viral proteins. HIV single-stranded RNA and proteins assemble beneath the host cell membrane, into which glycoprotein 41 and glycoprotein 120 are inserted. The membrane bursts out, forming the viral envelope. Released viral particles, complete maturation, incorporated precursor proteins are cleaved by viral protease presented in viral particles. Although CD4 binds to the envelope glycoprotein of HIV-1, a second receptor is necessary for entry and infection. The T cell tropic strains of HIV-1 use the co-receptor CHCR4, while the macrophage tropic strain use CCR5. Both are receptors for chemokines, and their normal ligands can block HIV infection of the cell. Individual isolates of HIV different in their ability to induce syncytia formation. HIV-1 isolates from different sources were classified as syncytium-inducing and non-syncytium-inducing. These differences correlated with the ability of the virus to infect T cells or macrophages. T-topic strains were syncytia inducing, whereas M-topic strain was not syncytium inducing. A region called V3 loop plays a role in choice of receptors used by the virus. Thank you.